go through my presentation, I would like to give you an overview of what we work on in my lab at UCLA. Basically, the focus of my lab is to form 3D hydrogel scaffold, which can have different properties, and we can tune those properties for different medical application. Once we form these 3D scaffolds, we combine them with different type of nanomaterials and different microscale technology to use them for different application. And my lab focus mainly on the use of these microfabricated biomaterials for soft tissue engineering application, or we can further modify the chemistry of this biomaterial in a way that they can adhere to the surface of the wet tissue, so we can use them as surgical bioadhesive and sealant for different surgical procedure. In the area of soft tissue engineering application, we have done lots of work on forming electroconductive biomaterials, which can be used for regeneration of hard tissue. Also, some of these formulation can be loaded with different type of therapeutic agent, cytokines, and even genes to form in a sprayable format, so we can spray them on the wound to control different phases of wound healing process. Also, these biomaterials can be used as bioinks to form 3D printed structure of complex vascularized tissue. In the area of surgical materials, we have done lots of work on forming elastin-based material that can adhere to the surface of elastic and soft tissue in the body. So we can use them to seal different defects on internal organs, such as lungs. We can use them for anastomosis. And also recently we developed formulation that can adhere to the surface of the cornea and can be loaded with different type of medication and drug, so we can use them to seal laceration and stromal defect on the cornea. We got large funding from Department of Defense, and there is a startup company, GelMedics, from the lab that working on further development of this technology to treat different ocular diseases. And some of our biomaterial can be also used for application where we have bone loss. We can use different type of nanomaterials, incorporate them in our bioadhesives, and use them for the treatment of different dental diseases, such as periimplantitis. Once we form these biomaterials with different function, we call them multifunctional material, we test them in animals, and we start with larger set of samples, or a larger set of formulation in small animals, and then through this test we screen a specific formulation that will be tested in large animals. And of course our goals is to translate some of this technology from the lab for human use. One of the biopolymer that we are very interested is elastin. As you know, elastin is a natural protein which is synthesized in our body during the development of different organs. It's known as a most persistent protein in the body. Basically, we don't lose elastin easily in our organs. However, during development of different diseases or as we age, we lose elastin in these organs. And one example of that is skin. When we lose elastin in our skins, we see wrinkles. So in the field of tissue engineering, we try to use different source of soluble elastin, such as animal-derived elastin or elastin like polypeptides, and even the topoelastin, which is the monomer that form elastic fibers, to form 3D scaffold, which can be used to regenerate these elastic organs in the body. So in my team, a few years ago, we start working on topoelastin. It is the soluble monomer that form elastic fibers in our body. Topoelastin has lots of great properties. It has many lysine residue in one topoelastin molecule. So when we expose it to the cells, it can interact with different cells and help regeneration. So we use topoelastin molecules and we modify it with a one-step reaction. Through acrylation, we form metacrelated topoelastin, it's short for metro, that has this functional group on the polymer chain. So once we expose it to the light, and if we add a photoinitiator, it can cross-link within a few seconds to form a highly elastic biomaterial. And we show that by changing the degree of the functionalization in metro, you are able to tune different properties of this biopolymer. And one important property for tissue engineering application, as you know, is mechanical properties of the material. And if you work with naturally derived material, although they have lots of great advantages, they lack elasticity and proper mechanical properties. With metro, by this modification, we show that the hydrogel are highly elastic and they can stretch up to 400% before we rupture them. And here are videos of mechanical testing. The top one is a tensile test, and this video showing torsion test on metro. And this video, I wanted to show you that this material has toughness, but also it is very elastic, and it is a great candidate for the regeneration of the soft and elastic tissue in the body. And with these panels data based on tensile test, we show that these properties of the metro is tunable basically by changing the degree of the functionalization or by changing the concentration of the protein that you use to form these scaffolds. You can tune the properties. And this opened lots of avenues for us to use metro for different applications, because we can not only tune the mechanical properties of this biopolymer, we can also control other properties, right? Degradation rate, swelling, and even biological properties of the metro when we interface it to different tissues for different regeneration application. And in our recent work, when I was in Boston at Northeastern University, I collaborated with a medical doctor at Harvard. And one application that we look into is how now that metro with this elastics, how we can use it not only for tissue engineering application, but also use it for sealing different defects on the elastic tissue following surgical procedure. And in this video, you see a pig line. We form a defect on the pig line, a hole, and once we observe air leakages from this defect, we seal it with the biomaterial. We modify the formulation of the metro to improve the adhesion properties of the metro to the wet surfaces. And here in this experiment, our indication was lying. And then we also purchased different type of commercial glues such as Porogel, Co-Seal, Evisel. Evisel is a fibrin glue. Co-Seal is a PEG-based glue, and Porogel is based on PEG linker and human albumin. And Porogel is the only FDA approved that surgery use it for lung surgery. And unfortunately, following the application of Porogel, the patients remain with extensive air leak, which can extend the day that they have to spend in hospital after they go through lung surgery. So our goal was to improve metro properties in a way that we can achieve higher best pressure, better adhesive properties to the tissue once we apply it. When we developed this biomaterial, it published in Science Translational Medicine, extensive characterization to find the right formulation. Then we applied in different model. And as I mentioned, we always start with a small animal model. So this work was in collaboration with a medical doctor at Harvard. And then what they did to test this material in rats, they formed a defect on the lung of the rat with biopsy. We deliver our metro as a liquid on the defect site. Topoelastin has this thermoresponsive property. Once it touches the surface of the tissue at 37 degrees, the polymer aggregates. And as we have this aggregation, or we call it coacervation, so it's confined to a defect site on the line. And then to further stabilize it, we use light to photopolymerize it on the tissue. And after exposing to light for less than a minute, we could form a very nice seal on the defect. And then following this experiment, we did burst pressure test. I put the scheme here for the burst pressure test. We connect the line to a syringe pump so we can apply air. And then there is a pressure sensor here. As the line bursts, we can measure the pressure. And this is reported as burst pressure. Here you see data for ex vivo. This is on dead animal. So we form the same size defect, seal it with metro and different type of commercial glue. And also we close the defect by suturing. And we show that for all these conventional method and even the use of the best surgical material in market, Porcine, the burst pressure was much lower compared to our optimized formulation of metro. And then after 4 days, once we treat the defect in the line with all these formulation, we kept the animals, and after 4 days, we sack the animals and run the burst pressure again. And what we observed for the MetroSeal line, the burst pressure was very close to the healthy line. Basically, Metro seal the defect, stop the air leakages at the beginning, but by time it also helped regeneration, the defect site that we form on the line. So to further confirm this data and then to see that if Metro can be used for clinical application, we will move on from a small animal to large animal. And this time our model was pig. So we form defect on the pig line, and then we spray our hydrogel, we photopolymerize it for a short period of time. And then here you see the defect on the line, and then this white part is a polymer that cross-link on the surface of the tissue. Once we seal the defect, we did ultrasound every other day to make sure that there is no leak from the line. And also after 2-3 weeks, we did lots of study to see that how the tissue heal. And we also wanted to assess the inflammation as we apply this material on the line. And then with this, we confirmed that we did experiment on 6 pigs, and our data show that we can have proper sealing and some degree of tissue healing and regeneration once we apply this surgical material on the defect. So this was done in collaboration with Beth Israel Hospital in Boston. And when we moved to UCLA, here I work with great colleagues at UCLA Medical School. And our next step with this project is how we can use it to basically seal larger defects under extensive bleeding with this material. And then we use different models like liver bleeding model, we use heart model, and we learned that in addition to adhesive properties, we need to modify the material in a way that it also have hemostatic properties. So we not only seal the defect, we also control extensive bleeding once we apply it on this on a different type of organs with different sizes of defect in the body. And this is our ongoing project at UCLA. Now I want to move on to another area in my lab that is still we use elastic biomaterials that has also adhesive properties to the surface of the tissue. But this time we want to design this material in a way that they also help regeneration of tissues that cells communicate through electrical conductivity inside those tissues. So basically, our goal was to form adhesive biomaterial which can be used for example, for neural tissue engineering application or for cardiac tissue engineering application. And as you know, there are many studies in the literature using different type of electroconductive polymer or nanoparticle like graphene oxide, carbon nanotube. They incorporate them in the biomaterial to form electroconductive scaffold. But the limitation with some of this technology is cytotoxicity. We have to limit the use of conductive polymer or nanoparticles to have this material electroconductive, but at the same time, we can use them for a tissue engineering application. So to avoid this problem in my lab, one of the PhD students work on how we can make this scaffold in a way that they can be electroconductive, but also highly biocompatible. So we went through different sources of component that we can use to give conductivity to our naturally derived polymer. And one category of this component is ionic liquid. Ionic liquid are organic salts that are liquid below 100 degrees. They have high water solubility and they have high ionic conductivity. So a specific formulation of bioionic liquid, it's cooling-based bioionic liquid, is also known as highly biocompatible. So for the first time, we use bioionic liquid, and again, through a one-step reaction with acrylic acid, we form acrylated bioionic liquid. Once we form this modified bioionic liquid, it can be combined with different type of polymer that are functionalized in the same way by mixing different ratio of acrylated bioionic liquid with any different biopolymer. Here, I put gelatin methacrylate or GELMA as an example. You can add photoinitiator, and by using visible light, you can form bioionic functionalized polymer that has conductivity, and you can finely tune the level of the conductivity in this biomaterial. So once we form this functionalized polymer, they can be used as an injectable formulation. You can directly inject them to any organs and cross-link it with visible light, or you can also use this formulation to form electroconductive and adhesive patches that can form as a prefabricated patch, and you can apply it on the heart after, for example, MI, to further improve the heart and have regeneration in the defect site. So I'm going to discuss a little bit about this approach that we recently published, and in this work we form electrospin sheet. We use electrospin techniques, and we use a biopolymer as an example, GELMA. We form this fibrous structure based on GELMA. To stabilize it, we soak it in a photoinitiator solution, and we add light to cross-link this fibrous structure, and then cross-link our bioacrylated bioionic liquid on the surface of this this patch to functionalize it with bioionic liquid. Once we add BioIL, the patch was ready, so we can load it with different type of cells, or we can even use it as a scaffold without cells. We place it on the heart, we cross-link it with visible light to form this patch adhere to the surface of the heart. And then with these fabrication techniques, we were able to tune different properties of the scaffold, including mechanical properties, was tunable by changing the polymer concentration that we use to form the patch, and also by changing the concentration of the bioionic liquid that we incorporate in our electroconductive patches. My student did lots of study in vitro to assess the electroconductivity of the form patch. One example is this ex vivo experiment where he extracted abdominal tissue and he placed 2 pieces of abdominal tissue on the surface of the conductive patch. He applied electrical current or force on the tissue on the left, and then he increased the voltage until he observed synchronized beating between the 2 pieces of the tissue that we placed on the patch. And then through this experiment, we calculate excitation threshold voltage, and we found that as we increase the conductivity of the patch we place underneath of the tissue, the excitation threshold reduced. Basically, tissue responds to the level of the conductivity of the patch that we engineer. To further explore this, in vitro, we extracted cardiomyocytes from rat hearts, and these cells were cultured on the biomaterial with different level of conductivity. One advantage with this technology is that we can finely tune the level of the conductivity in biomaterial. by simply changing the bioionic liquid concentration. For example, here you see that for different scaffold formed with different polymer concentration, as we add different amount of bioionic liquid on this scaffold, we can tune the conductivity. So with 100, we get highly conductive scaffold. By adding 66, it's average, and when we add 33% bio-IL, the conductivity is lower. So basically, we can make substrate with a wide range of electroconductive properties. We formed this substrate, we selected some formulation of that, and then we cultured cardiomyocyte on the surface of them, and then we measure the frequency of the cells, beat frequency of the cells over time. And if you look at this figure, you see that the cells respond to the level of the conductivity in the biomaterial. The blue line is the material with zero bio-IL. It is non-conductive scaffold. And the yellow line is the material formed with the highest level of conductivity. And you see that the beating frequency for both of them is lower compared to the formulation that has medium concentration of bioionic liquid. So when we wanted to understand better what happened when we have this different beating frequency of the cells, we did immunostaining for different protein expressed by cardiomyocyte over time. The picture on the top shows a substrate with no electroconductivity. So for this substrate, we did a staining for different markers, such as sarcomeric alpha-actinin, connexin 43 expressed by cardiomyocyte. And we show that as we have no conductivity, the expression of these proteins is lower compared to the formulation that we introduced bioionic liquid, and it has a higher conductivity. And here are quantification data showing the expression of connexin 43 is higher in electroconductive biomaterial. And basically, connexin 43 is very important protein that shows the function of the cardiac cells in terms of cell-cell communication, and therefore the beating characteristics of the cardiomyocyte. And in these videos, I show that on the left side, these are the cardiomyocyte culture on non-conductive patches. You see that they are beatings in different areas of the scaffold. However, as we make these materials electroconductive, we have synchronized beating characteristics of the cells as we culture them in the biomaterial. So there is a benefit in improving cell function as we work with electroconductive substrate. This is, I'm not going to confirm with our research team, but also there are many other research teams focusing on this aspect, and in agreement with those studies, electroconductive biomaterial can further improve the function of the cells. So our next step for this project is, now that we can finely tune the electroconductivity in our biomaterial, we want to look into how they behave when we put patches with different conductivity to the heart, and then what happened in vivo. But before we do this experiment, we also did some adhesion tests in the lab. On the top, you see wound closure test. My students formed a patch, they stick it to the heart, and as we add bioionic liquid to our formulation, it can form electrostatic interaction with the tissue. And we also add light, it also chemically bonds to the tissue, so we can achieve adhesion. And we show that by adding more bionic liquid, we can have better adhesion on the heart. In the bottom panel, you see the burst pressure test. This is whole rat heart. And then we show that the patches can adhere to the heart. So if when you apply it, you do not even to suture it to make it stable on the heart. So here are some preliminary data that we did. And this is the rat heart. We put the patch, we photopolymerize it with visible light, and in this video you see the patch after 3 weeks it remains stable on the surface of the heart. And with our functional data, we show that as we treat the heart post-MI with conductive patches, we have better heart function compared to the non-conductive patches that we use for this experiment. These are some preliminary data, but now with in collaboration with Professor Ardehali at UCLA, we are trying to do a large, larger scale of in vivo test to understand exactly what is the effect of electroconductivity once we implant these samples in animals. So as a last part of my presentation, I also move on to another recent area that we start working on, and it is based on the use of some of these elastic formulation that we have in the lab as bioink to form complex 3D structures. And in this project published in Advanced Materials a few months ago, my students focused on engineering vascularized heart tissue, and her goal was to use and design two different bioinks. One bioink was based on gelatin. As you know, gelatin is denatured collagen, one of the most important extracellular matrix protein in our hearts. And she used acrylated form of gelatin, gelma, to form cardiac tissue. And she also used an elastic material in the lab. For this project, she used metro to form blood vessel. By optimizing two different bioink and loaded them with different cell types for engineering blood vessel and cardiac tissue, she used our two-nozzle bioprinter and fresh printing techniques to form a structure with vasculature with different sizes in a construct that can be used for heart tissue regeneration. So basically, before we form complex structure based on these bioinks, we did lots of experiments to basically optimize the printing condition, including extrusion pressure, printing speed, and the goal of this experiment, also controlling the temperature. The goal of this experiment is to form a structure with better resolution. And as we optimize this condition, we were able to form multi-layer structures based on our bioink. Basically, we can combine this through layer by layer assembly. We can control the thickness of the structure that we bioprint. So we were able also, by using this fresh technique, we can use any type of bioink that has a soft mechanical properties. We can form complex structure based on these biomaterials, as you see in these images. So one example to show that the design bioink are biocompatible, we form a multi-layer structures. In one layer, we use a gelatin-based bioink laden with cardiac fibroblasts and cardiomyocyte freshly isolated from rat heart. And for the second layer, we use our second nozzle that was loaded with elastin-based bioink laden with endothelial cells. And then as we form this multi-layer, liquid in the bath, and then after we form this structure, we remove it from the bath and we place it in culture. And here in these images, we show that the cells, different cell lines incorporated in different layers, they are all viable. We observe high cell viability, and also the cells express the right proteins. For cardiomyocyte, we did a staining for sarcomeric alpha-actinin, so the cells express this protein. And for the endothelial cells, we did a staining for CD31. Moving from that, we also look into how we can form more complex structure. For this experiment, again, my students use a two-nozzle printer. One nozzle was loaded with metro, laden with UX cells. And for the surrounding matrix shown by yellow color here, she used gelatin-based bioink loaded with cardiomyocyte and cardiac fibroblasts. And then as we form this structure, you see that it's a continuous process. We can 3D print these structures in a short time, photocross-link it in the bath, and once we form our structure, we can take it out from the bath and then put it in culture. And here are some data showing the viability of the cells that we incorporate in different parts of the construct. This is the vascular part formed by our elastin-based biomaterial, showing that the cells are viable, and also in the surrounding matrix that we incorporate cardiomyocyte and cardiac fibroblasts, we observe cell viability, high cell viability. We also, similar to the previous in vitro testing, we did a staining for different cardiac markers and also for CD31 to ensure that the cells that we incorporated and use it to form this scaffold, they are all functional, and also they have right phenotype. One limitation of 3D bioprinting is that for the sensitive cells, we are able to incorporate them in the printed structure. They are viable But in many cases, because of the printing process, it can be long process, or due to the shear stress we put this on the cells will not have the function that we want. And this applies more for sensitive cells like cardiomyocytes. So one characterization that we did, we measured the beating frequency, or the beating of these cells over time. And as we show this video, even after day 15, all the cells that we incorporate in the surrounding of the vessel that we form, they beat in circoronies. So we have right characteristics of the cardiomyocyte once we 3D print it into this, like, complex structure. So I think my time is over. With this, I would like to acknowledge my students, both at Northeastern and here at UCLA, who worked very hard in the lab, even during COVID. We have our lab running and the students worked very hard. All my great collaborators, and also my research funding from NIH, American Heart Association, and DoD. Thank you very much, and I'm happy to answer your question. And of course, this is our beautiful campus at UCLA, and I welcome all of you to visit us one day, hopefully after pandemic.